record this. Thanks everyone for coming to our Open Sci Tech webinar. My name's Megan. I'm from the IEC, from the Hive IEC group. So thank you for joining our Open Sci Tech webinar. We are going to talk about our new HuBMAP tools, which if you're on our email list, or if you've heard about this at all, we have a bunch of new tools on the HuBMAP data portal, as well as some new organs, assays, lots of new data available as of recently in the last couple of months. So we're very excited to have two presentations today about two of our new tools. So just quick housekeeping note, if you've been to one of these webinars before, you'll be familiar with this. We're just a reminder for folks to put any questions that you have for speakers into the chat window, into the chat section, and those will go to our speakers who can answer those during Q&A when we have time for that. And so to introduce our speakers, we've got Mark, who's from the HuBMAP Hive Tools component at Harvard Medical School, and he'll be talking about exploring spatial single-cell data with Vitessce visualizations. Vitessce is one of our new tools. And then Andreas and Bruce, who are from the HuBMAP Hive mapping component at Indiana University, and he'll be talking about registering your tissue so that it can be explored in context, explaining the new releases of the CCF registration and exploration user interfaces. So two great presentations today, and we will have Mark kick us off. Mark, have you joined us, and are you ready for your presentation?
Yes, so I can start sharing screen.
All right, awesome. Take it away, Mark.
Thank you. Can you see this?
Yes.
Okay, great. So today I'll be talking to you about a single cell data visualization tool called Vitessce. I'm Mark, and I am a graduate student in the Gellenborg lab as the Hive tools component at Harvard Medical School. So for some background, the HuBMAP TMCs are generating data sets at single cell resolution, which span multiple modalities, including transcriptomics, chromatin accessibility, imaging and microscopy data. And then as this data submitted, Hive processing pipelines to generate derived data sets, which may include dimensionality reductions, cell segmentations, and then clusterings or cell type assignments. And so there are currently many tools for visualization individualities, such as genome browsers or image viewers or scatterplot viewers, but few tools which enable visualization of all of these modalities simultaneously. And then, as we can see in this plot taken from the data portal homepage, the HuBMAP TMCs have already submitted like hundreds of data sets across tens of modalities. We would like to provide web-based interactive visualizations for all of these data sets, which required us to rethink the existing visualization approaches in terms of both human-computer interaction and software engineering. And so to address these challenges, we developed a framework for interactive visualization of single-cell data called Vitessce. And you can see the screenshot here. And Vitessce can be used as a web-based tool. So this is a screenshot of Vitessce in a web browser. And Vitessce supports many visualization types, including spatial or imaging data with cell segmentations, as we see here in the center left of the screenshot. And then on the right of that are some scatter plots with multiple dimensionality reductions, as well as a heat map component, a violin plot for gene expression data. And so Vitessce was designed to be modular, so that different types of plots and also controller components included the data and measurements that the experimenter has. And so its data files does not require any specialized server. So this is intended to reduce infrastructure costs and make it easy to deploy the tool. And so to enable relation of things like cells and genes and cell types across the plots, Vitessce contains a flexible coordination framework. So plots can be linked on these data properties or also visual properties like color maps, image channels, and things like that. So in addition to being a web application, Vitessce can be used as a Jupyter widget in Python and as an R HTML widget or Shiny widget. And Vitessce can load data from and data objects that are saved as ZAR stores. So this should simplify the data conversion process for certain single cell data structures. So we built Vitessce with custom visualization components, but also where we can, we leverage existing open source visual libraries. This includes HuBMAP for visualization, Viv for loading and rendering 2D and 3D imaging data, and Vega Lite for statistical plots such as bar charts and violin plots. So Vitessce can be used not only as a standalone web application, but as I mentioned, as a widget in Python or R, and also as an embedded component in other web apps. So this is how Vitessce is used in the HuBMAP data portal. And so I'll use the HuBMAP data portal to show some examples of what Vitessce can do. With the modular architecture, it can support modalities including autofluorescence microscopy, light sheet microscopy, transcriptomics, chromatin accessibility data sets, and then Codex data sets. So first, I'll show an example of Vitessce being used in the data portal with an imaging data set. So this is data autofluorescence microscopy from a kidney. And so here Vitessce is loaded data directly from an OME TIFF file. And the image has 3 channels, and we can use the image layer controller to change those dynamics, the color mapping. We can add channels and change the range of the color mapping. And so Vitessce supports pyramidal image files. So as the user zooms, higher resolution data is loaded, and we see the more detailed. So in addition to 2D data, Vitessce puts 3D data through the Viv library that we use that's integrated into the component. So here, using a light sheet microscopy data set from a lymph node, we can check out this feature. So once we select using this dropdown, we change from a 2D rendering mode to a 3D rendering mode. The data which was first loaded as a 2D slice becomes a volume rendering. And so this takes a little while to load the extra data volume, but once it loads, we see the volume, and we can use extra control volumes, and we can use like a maximum intensity projection as we see here. And we can use these sliders to clip the volume along the X, Y, or Z plane or axes. And so we can also change colors as we do in 2D. Yeah, so this is zooming and panning around the 3D volume. So for something like transcriptomics data set, we can switch out the components that we include in visualization. So here we have an RC data set, and we see there's a scatter plot and a heat map. Also a violin plot that we can use once we select a gene. And this shows the normalized expression values across each of the clusters. We can add labels to the scatter plot. And an interesting gene here is CD79A. And we see this is expressed highly in clusters 0, 1, and 9. And so this gene is expressed in B cells. So, yeah, we don't have cell type annotations here yet, but we can see just clusters that are from an unsupervised algorithm currently in the portal. And then for Codex data sets, we can add an image data plot, a scatter plot, and a heat map. So something special about the Codex data that we have in the portal is we have image bit masks for cell segmentations. So these are, we render these in like a multi-scale fashion. So as you zoom, the bit masks become higher resolution. And like before, we can select an antigen in this case, and then the segmentation mask colors will be mapped to the quantification for that antigen. So here we select the pan-CK, and if you'll notice, the scatter plot here contains over 70,000 cells, and we recently implemented a feature for dynamic opacity settings. So this prevents overplotting in the scatter plot, and it makes it easier to see the structure in some dense clusters. For single cell attack-seq data, we can use Vitessce for the chromatin accessibility data with a genome browser track visualization. Here, this is also multi-scale. As we can load more detailed data for the chromatin accessibility as well as the gene annotations. You'll see more genes will appear, and the scatter dimensionality reduction can be linked to the genome browser tracks. So as you select a cluster, it can be highlighted in across the visualizations. So quickly, I will walk through how to create a Vitessce visualization for a local data set. So we wanted to create a wrapper for Python because bioinformaticians typically have local data and they wanted to analyze this using a Python or R packages. And so it's easiest if the visualization is also in this same ecosystem. So for the Python and R packages, the main features are local data loading functions. So these help you load data stored in commonly used single cell data structures and file formats. Functions for configuring the grid of visualization components. Functions for coordinating parameters across multiple linked visualizations. Interactive widgets for Jupyter Notebooks, RStudio, and our Shiny apps. Functions for helping you to export your data and the visualizations to the cloud using app storage systems such as AWS S3. So in Python, the package currently supports loading OME TIFF images, as well as ANData, Loom, and Snap Tools objects. And then in R, the package supports loading OME TIFF images and Threat objects. And we're actively working on functions to load data stored in other commonly used single cell file formats. So the first step to configure the visualization in Python would be to import the functions from the package. And then we a new configuration and match an object in our single cell data. Next, we add the visualizations that we are interested in using. So in this case, we might add 2 scatter plots. So we specify that the data set that we want to use is the one that we added here. We want the scatter plot component, and then some extra parameters in this case would be to specify the dimensionality reduction that we are using for the scatter plot. We might add the cell set manager component and the gene manager to select cells and genes. And we can add a heat map visualization, and then we can use like syntax in Python to lay this out in a grid. So the slashes are horizontal or vertical concatenation, and the pipes are horizontal concatenation. And you'll see this line here, once you lay out the plots like this, it will there will be the 2 scatter plots stacked on top of each other on the left, and then the other plots on the right with the heat map on the bottom. And as an example, so that same code in a notebook, you'll see here. And once you run the widget, the widget will load, and we can interact with the widget and select a gene. So this is a blood data set. So if we select the same CD79A gene, we'll see that it's highly expressed here in one of the clusters, this one in yellow, and we can change the color mapping function to see it a little bit more clearly. So you'll see in red now, that cluster has high expression of that gene. And so once created your widget, you might want to export it to the cloud, and you can do this using the functions in the package. And you can even create a URL to share with your collaborators or to save for yourself. And then to learn more about Vitessce, we have installation instructions and example notebooks at vitessce.io. Some features that we have planned are selection of multiple genes, and then plots for visualizing this multiple gene selection. For instance, the commonly used dot plot. We would like to have better support for analytical backends. So when it differential expression in like a server side type of way, or you want to connect Vitessce to some machine learning model. So we currently don't have much support for this, but these are some use cases that in the future we would like to support. And then comparisons. So maybe you have 2 data sets, or you have a healthy and a disease type of data set, and you want to more easily compare or directly compare. So maybe you have a plot with better color maps or things like this. And so finally, I'd like to thank the other members of the Vitessce team, as well as the whole Gellenborg lab and the HuBMAP consortium for all of the support. Thank you. And I will check in the chat. I think there's a question from Martha Campbell Thompson. Tissue, was this maybe for a demo?
Hi, yes, it was I think the Codex slide you were showing. I didn't hear what the tissue type was.
Okay, so for Codex, that was the thymus. So I can paste the link to this example in the chat as well.
Thank you.
All right, thank you, Mark. Does anyone have any other questions? You can always feel free to drop questions for Mark into the chat. Thank you, Mark, for that great presentation and demo. We will move into our next presentation, which is Andreas and Bruce, if you guys would like to share your screen.
All right, here we go. Can everybody see my screen and hear my voice?
Yep.
All right, hello everybody. It's good to be back here, giving another demonstration of our tools. Naturally, these are not new tools. We just rather have developed these tools over the past couple of years and just are presenting new iterations every now and then. So today this is going to be very much demo focused, but we decided to leave a large portion at the end for Q&A, just to support any questions that attendees here might have for Bruce and myself. I want to start with this slide that some of you might have seen in the steering committee call just a few minutes ago, where we talk about the RUI registration contest that is running until September 30th, so until the end of the month, where you can add RUI registrations to existing registered tissue in the ingest portal. And we will go over how to do that in just a second here after the RUI demo proper. All right, so now I'm going to go and start with the RUI demo. Let me just switch over to another tab. Someone, Ellen or Bruce, if you could please grab the link here for the RUI and just pop it into the chat, that would be awesome. So this is going to be a little demonstration of how the registration user interface works. So as I go to this page, you can see this is the standalone version of this registration interface, which is available to anyone with an internet connection. So as you come to the page, you are presented with this modal, that is this little window here that you have to basically go through before you get to the actual application. So I'm going to type in my name. And then I choose the donor sex. In this case, let's go with male. And then I can select an organ from this organ carousel. Let's go ahead and use let's go with the large intestine. And then we can start the registration. Then you can see that the 3D stage here in the middle gets populated, in this case with the male colon model. This colon has been modeled on the visible human data set. And then you will see that the RUI has 3 panes. So on the left, we have the anatomical structures pane. In the middle, we have the 3D pane or the 3D stage. And on the right side, we have the tissue block manipulation pane. So the anatomical structures pane gives us information about, as you might imagine, the anatomical structures that are present in this 3D model here. So we can see we have the ascending column, the cecum, the descending colon, etc., etc. And you can see as I hover over these icons that these sliders appear that allow us to set the opacity of the 3D model. You can see currently all of this is being set somewhat transparent at a 20% opacity level. I could just go ahead and slide that all the way up, and then you will see that the ascending colon here becomes 100% opaque. If I click this reset button, it goes back to the default of 20%. I can also turn it off completely. That is something that you would probably not necessarily do for the colon, but for organs with a lot of internal structures, like for example, the heart or the kidney, it is very useful to turn individual parts of the organ on and off. I'm going to turn this back on for now. So keep these anatomical structures in mind. They're going to become important in a second when we do the actual tissue registration. Next, we have here the so-called common extraction sites. And these common extraction sites are sites that we identified as important visual landmarks with experts. So currently for the colon, we have 4 of these or 5. So I'm going to turn these on real quick. Okay. The HuBMAP colon extraction sites are just a superset of these. So I'm going to turn these on. And then you will see here in the middle on the 3D stage that we suddenly get these green overlays. So these have been reported to us as important landmarks when extracting tissue. So oftentimes, for example, a team will extract a tissue mostly from, let's say, the transverse colon. And so this way I can colorize the transverse colon so that I know where it is in this particular organ model that I look like. Obviously, every individual human organ is different from each other. And so having these landmarks kind of helps with understanding where you are with regards to this reference organ here. And then I can also set the opacity here. Let's set this all the way up just so you see what it looks like. Here we go. All right. So now to the actually really interesting part, that's the 3D interaction with the organ. So organ sits here comfortably in the middle, and you can see here on the right side is a representation of a tissue block. And currently this is set at a volume. This is actually a cube of 1 cubic centimeters. So you can see the tissue block dimensions here are 10 by 10 by 10 millimeters. Let's go ahead and resize that real quick. Let's make that a little bit bigger. So let's make that maybe 2 centimeters by 15 centimeters, give it a depth of 25 centimeters. Obviously, I'm not a biologist. I don't know if that is a realistic size for a sample, but let's just go with that for now. So then you can see as I hover over this tissue block and I click and drag my left mouse button, I can move it around. Along the X and Y axis, you can see that the coordinates here, you can see how they change as I move the organ around. And it is currently locked with regards to the Z axis. So that's why the number doesn't change here. All right. A few more words before we do the actual registration about how I can adjust the view here. So you can see I can rotate my camera around the entire organ by using these radio buttons. So we got the anterior view, the right view, and the posterior view, and the left view. And that allows me to see the organ from all the different angles. So let's go back into anterior view. Let's say we want to register here into the transverse colon. I just drag this over here. And then, of course, the tissue block kind of disappears. So I'm going to turn the opacity here significantly down. And then I see it lying right in there. And then maybe let's go to the left side. Let's say we want to adjust the height. Here we go. Okay, and so now it's the time to actually adjust the rotation. We adjust the position or let's do the rotation. So to do that, we have this 3D preview mode here. So if I click this toggle switch, I now get into this camera mode where I can adjust my rotation freehand. Okay, and I just see that, oh, this is actually not inside of the organ yet. So I'm going to go back into registration mode. So maybe go to this side and then move it a little farther here. Here we can, by the way, here we can see the extraction side again. All right, back into 3D preview. And now I can see I'm nice and snug inside of the transverse colon here. And then I can use these sliders here to adjust the rotation of the block. Just for demonstration purposes, let's rotate it around a little bit. Here we go. All right, and then one very neat thing that you will see here is that we have these anatomical structure tags, okay? And currently it actually does say transverse colon. I'm gonna go back into registration view real quick, into the anterior view. I'm gonna lift this out real quick. So what you can see here is that now these fields here are empty. However, as I move my tissue block in there, it gets populated with transverse colon. We are using what's called 3D collision detection, which is exactly what, well, the word implies. We prompt for collision between two objects, two 3D objects. This is a feature that is being used all the time in video games and Hollywood movies, whenever you have any kind of collision between objects. So that's how we annotate your registration in addition to capturing position, rotation, and size. All right, so once setup is done, you can review and download your data. Okay, so this is our registration review. And then you can download your registration data as a JSON file. All right, I'm looking at my timer here. All right, doing good on time. All right, so that is the part about the RUI. And another important thing that I would like to cover is, so we have these common extraction sites here that serve as landmarks. However, another really neat thing that you can do with the RUI is you can reuse existing registrations. So I talked earlier about how I could just review and download the data that I just registered. And the format for this looks like this. Here we go. So this is the a JSON file that you actually generate whenever you download a registration. This comes from a collaboration with the team at GTech, which I'm going to talk about more in a second. So what you see here is first a whole bunch of boilerplate information. Then we have the creator first name and last name. With the data that I just generated, that would be my name. Then we have CCF annotations. As you can see, these are strings that contain URLs to Uberon. So these are Uberon terms for these anatomical structures. We have them. We have a crosswalk between the anatomical structures in the RUI and these anatomical structures on Uberon. And then you have all of this important spatial metadata that that make up our registration essentially. And so really important here is this, this key value pair here with placement. And then we have this RUI location object. Okay. So we have, for example, here the X, Y, and Z rotation, and we have the X, Y, and Z translation. And we also have the sizing, which is up here, X dimension, Y dimension, and Z dimension. All right, I would now like to demonstrate how you could reuse existing, how you can reuse existing registrations. Okay, so I'm gonna click on refresh here. And now I would like to show you a extraction site or a template that we registered with the GTech team a couple of months ago. So I'm gonna type in my name real quick, get past the modal screen. This is again for the large intestine, right? So then I can click on upload previous registration data, and I select an existing JSON file, in this case, this one here for GTech, okay? And then you will see that the 3D stage is automatically populated, and you see that the little tissue block automatically jumps in this predetermined position. And if we have another look back at the file here, you can see we have the X, Y, and Z translation, 112, 301, and 148 millimeters. And then if we go over here to the RUI, we see also 112, 301, 148 millimeters. And of course, the rotation has been adjusted as well. So then if I go into my 3D preview, go in here, and then I see a little tissue block sitting right there. And then, of course, you can reuse this, this JSON file as many times as you need. So specifically in this case, we are working with the GTech team to register a total of 1,918 tissue blocks from just a few templates. So we created 6 templates so far for the male reference organs. We're going to just copy those over into the female reference organs. And just doing these few registrations then allows us to register almost 2,000 tissue blocks via this kind of template method that we are currently working out. So that is one of the latest developments surrounding the registration user interface. All right, I want to come back one more time to the contest before I turn it over to Bruce. For this contest, the IAC is allowing everyone to add RUI registrations to already published data. And in order to really document how this is being done, we updated our SOP, and maybe someone on the call, Bruce or Ellen, maybe you can also take the link to this SOP and post that really quick into the chat as well. So this SOP, Standard Operating Procedure, is kind of like a manual for how to use the registration user interface. It comes with plenty of visual material, including, I'm a very big fan of screenshots, annotated screenshots. It comes with all of these screenshots. Another thing we added is we have now flowcharts that summarize the most important steps of using the registration user interface in just kind of a visually more pleasing and summarizing way. And then we have this long write-up here about how to access the RUI from within the HuBMAP ingest portal with 2 cases, registering a new sample with a RUI location. And you can see we added all of these annotated screenshots that tell you exactly how it's done. Of course, we have to update this SOP whenever there's an ingest portal update as well. And then we also outlined how you can add a RUI location to an existing sample if it's unpublished. And then here we have the special case. For a short period of time, you can also add a RUI location to published unregistered samples. And if you have any questions about these processes, please feel free to reach out to me. I'm going to put my email address here into the chat in just a second. All right, let me look at my notes. I think that pretty much covers it for my part. I'm now going to turn it over to Bruce, unless there are any questions right now. So let me open up the chat. Doesn't look there is anything yet. So I'm going to turn it over to Bruce now.
Okay, great. Thank you. Let me go ahead and share my screen. So Andy has been talking about the registration side of the CCF. I'll be talking about the exploration side of the CCF. And yeah, just a reminder, so for people in HuBMAP, use the ingest portal for registering your tissues. And we have, like I said, we have that contest open until the end of the month. And I'll show you where that eventually falls out at. So before I jump into doing a demo, I wanted to just talk about some of the new features that come out in the last few months. Actually, the first one that actually was last Friday. We have a greatly improved performance. If you've used the exploration user interface before, the loading takes about 10 to 20 seconds, depending on your computer. And so now we've got it, the load times are much faster, sometimes even up to just a couple seconds it takes. So it just gets you into the EUI a little bit faster, which is a good thing. And the second thing is we've refactored the RUI and the EUI as web components. This allows us to embed these user interfaces in different contexts with different consortia and in different languages. We write the RUI and the EUI in Angular, and both the ingest and data portal are written in React, and they're able to be used from those portals. And also over the summer, I was in a codeathon, and we created so I used that same web component to host Spark data and HuBMAP data in the common 3D space, the CCF space. So you could actually, I was actually able to load custom data from a totally different consortia, show it in the EUI, and even load in a reference organ that they had created in their own context. So, and a similar thing, we have sort of a data interoperability via this JSON LD format, and we've had this for a while, but both Spark and KPMP data, not all of their data, of course, but just some samples from those two consortia are in the EUI proper. And then we added Google Analytics and telemetry for our data for both the sites, so that we can start to understand what people are using, what they're not using, and sort of to help drive where we go. We're a data-driven organization, so this just allows us to get that data to help create an even better user experience. And over the summer, we opened up support for 11 different reference organs, which you'll see here soon. And more data is pouring in. Over the last few days, I've seen the numbers start to go up on the EUI. So that's a really good thing. So the first thing I'll show you is how to get to the EUI. There are other ways to get to this, but the primary way you can get to it is through the HuBMAP portal. If you look under Atlas and Tools, you see the RUI. This goes to the standalone version of the RUI that Andy showed off, and then you can click on the EUI. We also have the CCF portal, which sort of is an informational page about all things CCF. The ASCT plus B reporter is another effort that we won't be talking in too much detail today, but that is generating the semantic space that we're working in, deriving these for each organ, the anatomical structures, cell types, and biomarkers associated with that organ, relying heavily, of course, on the different Uberon ontologies HGNC, cell ontology, etc. But working with over 100 SMEs to really dial that in. So let me show you the EUI. So you have this initial load. Our methodologies, we actually query the search API in HuBMAP and say, give me all the tissue samples that have been registered with the RUI, and then on the fly we convert that to JSON LD and load it into our semantic triple store that we use for as our client-side database. If you've been around HuBMAP for a while, I'm sure you've seen this screen before. I'll show you, first of all, on the left, you have the anatomical structures. This is the AS from the ASCT plus B. These were pulled from version 1.0 of that. And if you open them up, you can see the different, it's a partonomy, so the different parts of each different organ. And you'll see the ones in bold have actual samples associated with it as determined by your RUI placements associated with your sample. And then in the middle, obviously you have the two referenced human bodies, male and female. On the top, you can change the organs that you would like to see. And we also have an information display. So there are 4 tissue blocks associated with or registered with the large intestine. And some new ones that actually came in. I'm going to go ahead and turn them on since we got them. So we got 16 thymus samples reselling. So that's great. And then on the right is first of all, we have a listing of all the aggregation number of tissue blocks, 104 tissue blocks. I think we were at something like 80 or 53 before we started this big push to get more tissue data. So that's clicking along. And then on the right, this is a listing of all the donors that are matching our current filter, which I'll get into over there. And so you can turn on, usually what we do is we turn on a few of these. So you can, okay, so this one, Vanderbilt is a kidney. So as you zoom in, these tissues become more, or the organs become more transparent. I'm gonna turn off the large intestine here. Let's see. Maybe I'll just turn one of these on. That'll be easier. This is linked, so there it is. So this orange block here, 56 female. So what you see underneath, you have the structure is donor, tissue block, tissue section. Data sets can fall at either the tissue block or the tissue section level. And you'll notice that you can click on any, so I can click on a donor, and this opens up a little frame that shows the data portal for that specific donor, and then you can switch between the two. So now we have the donor information exactly as you would see in the portal, and you can always then open that up in the portal. There it goes. And you can quickly then jump into a tissue block or tissue data to see that information. This shows, so within a tissue block, you have multiple tissue sections. Each green line is a tissue section. This visualization, right now, we don't have really good data as far as tissue section ordering and thickness that we've been able to put into here, but over time this will get a little more sophisticated. But essentially, this shows the sections in that tissue block, and the data is the data on associated with that tissue. In this case, it's this with the tissue block, but others are with tissue sections. Let's see. So then the other thing I wanted to show is the filter. So we can filter by sex, age, and BMI. If I switch it to female, it'll just show the female body and the associated data. There you go. So here's and you can filter by assay type and tissue provider. I wanted to do one thing is to remove the non-HuBMAP. So we have KPMP and Spark data that's been loaded in here as well, but that then limits it to just the HuBMAP data. So you can always go and update with that. Let's see. And the other thing I should note is this is showing the published data, but if you log in, it can also show the unpublished data if you have the rights for that. I just wanted to quickly show one other thing. We also created a web component that lets, for the organ specific pages, which are coming out pretty soon, we created a web component for them to drop into their React-based application that just gives you a quick, so you can load up kidney and it shows all the tissue blocks in there. You can switch between male and female, and it has like the information and then links to relevant information for the CCF. Okay, so at this point, I think I'll open it up for questions and answers. I think I saw something on the chat. Okay, so what determines the angle of the tissue block within the organ? So when you register with the RUI, you say, you set the orientation. And of course, then the reference organ is then placed inside the body so that it might rotate it further. But there's those placements that Andy showed earlier with the rotation determines those angles. Are there any other questions? I'll open up for any questions about the RUI.
So Andy and Bruce. So when you have several blocks that come from different donors, how do you know what's the actual overlap between them? I'm assuming that the placement is a little bit approximate, right?
Yeah, I mean the placement is, well, the placement is the best effort. Like obviously, we're not going. We're hoping, yeah, with the R2 UI, we're hoping to use the data associated with that sample to create an even better placement. So we can look at the landmarks in the data.
Yeah, so that was why I was going to ask whether you can have a more detailed level where you have anatomical landmarks that you can use to match with the histology sample.
Yep. Yeah, that's something we've been working on, but not quite ready yet. But that is in the works right now. So.
Yeah, okay, because that's also interesting or important to know whether we have enough coverage of a certain area of the kidney but not of another one, and I'm still not sure, are they all from the same place or not? Yeah, thank you.
So going forward, we will actually know cell type populations per anatomical structure. And what you can then do is you can basically fill out these anatomical structures with the correct cell type population. And that has been done for mouse brain. You might have seen the EPFL blue brain mouse brain browser. And based on that molecular data, you can then do this so-called stage 2 registration. But within HuBMAP, we don't have that kind of data yet. It's coming.
But
into
I think there is
very
an intermediate, Katya. I think there is an intermediate level that it's the pattern, the level of the tissue. So before you go to single cell, you go to the anatomical structures that they form, the groupings of the cells and things like that.
Absolutely. And as you know, we are in contact with several of the TMCs now to get whole slide images for the next Kaggle competition, which is ultimately hoping to develop an classifier for different types of tissues. So I think that also is in support of that approach. But I think we might be fast enough to actually do the cell type populations per anatomical structure. I think Rahul has made major progress, and there's now data sets that are coming into existence that we can use to do that for specific areas. Right now, we don't have too many data sets that have the same spatial location. We do have 10 tissue blocks for the kidney that have the same relocation, and then we have another set of 10 tissue blocks that also have a different relocation. But other than this, they are all quite different in terms of size and spatial position. And
so if I understand correctly what you say, it's just allowing you to know if you are going in the same location, that the block goes in the same location as the previous one, because you need some sort of golden standard to know that even from the beginning, they were attributed to the correct location.
Yeah, you have to have biomolecular data for the stage 2 registration, not just for the tissue blocks that you are trying to register, but for the organ part into which you try to register into.
Yeah. Thank you.
Great question, yeah. Niels has a question about a circular logic if we use molecular information for placement. I think it's very similar to the Artemis references, right? But the Artemis reference doesn't know we won't get in terms of cell type annotations. However, there is continuous effort to create better and better references. And similarly here, we are in the process of using better and better data at higher and higher spatial resolution, and also different types of assays to get better and better references. But yeah, Niels, this is always an issue. If you have a map, you map to that particular reference map.
Maybe one thing I want to add again, just to come back to the contest that is currently going on, that I wanted to remind everybody about. If you have concrete questions about how to add re-registrations to the data that you already entered and you can't figure out how to do it, please let me know. I put my email address into the chat earlier. This is a workflow that none of us has done before, just because published data was not editable so far, but it is right now. So if you have any questions, please let us know.
And yes, everything is versioned. So there are ASCT+B tables, and then there are associated 3D reference objects for anatomical structures, major anatomical structures, and they go together. So there's a crosswalk or mapping between the anatomical structures for which we know the 3D outline and for which we know the name. And as you know, an atlas ultimately is a definition of different areas that have an agreed upon name. And the agreement on names and areas is non-trivial. So there are now 150 experts across 16 consortia collectively working on these tables and on the reference organs. And it's an information science challenge, I would argue, to get that organized, but also to have a community consensus emerge, as opposed to having a bunch of war zones, which you don't want.
And as far as versioning of the placement, the mapping, the RUI locations, their metadata on the sample. So, and the samples themselves theoretically are versioned, but as it is now, once it's published, it's not supposed to be. It's not changeable. Only for a short period of time it is, but yeah.
I think going forward, what you really want to do is to select all the tissue blocks in the upper part of the kidney, all the tissue blocks in the lower part of the kidney. And then what you want to do is to look at their cell profiles, the cell census for each. And you want to see how that changes as age increases or BMI increases or decreases over time. I think these are the kind of comparisons that many seem to be wanting to do. So what we can now do is to support not only spatial search, but also search based on these additional attributes in comparison across tissue blocks and their data sets. And so,
but Kati, that's where variability comes. If they are not exactly from the same location in the kidney, you don't know if the variability is because of location or something special about the donor.
Yeah, so right now the current RUI is accurate plus minus, what was it, Andy, you run this user study.
1.32 millimeters.
And that's not good enough. So that's why we are working on the R2 UI, which absolutely requires that you have biomolecular data for the reference itself, which we really don't have in HuBMAP. That's why we are collaborating with other efforts. But right now we trust that that those which register tissue blocks do the best effort to get it in the right place. And then what you have right now is just kidney. And many other efforts out there, they would report cell by gene matrices for an organ, not even for a very specific part of the organ. What we will have in HuBMAP is a much more fine detailed registration. We trust those which extracted the tissue to register it back in. And that gives us a better understanding of the spatial size and position and rotation of the tissue block. Keep in mind that you now also have information on how the tissue was cut, how these sections sequence and align to each other. And very soon we will also have 3D data. You saw in Niels's presentation a 3D data block that now exists from the light sheet data that the University of Florida provided. So I think we all would like to have more 3D data, and I think it's coming into existence.
All right. Great stuff. Thank you for all those questions and that discussion and for some awesome presentations. I think we're just about out of time. So I think there's some follow-up and lots of links in the chat if anyone wants to check those out. And as everyone knows, all of these tools and resources are available on the data portal, which is available to everyone. So please go ahead and check those out. Now you should have a better idea of what's available to you. And thank you everyone for joining us.
Thank you for organizing. Bye.
Thanks everyone. Bye-bye.